Welcome to this microbiology tutorial on culture media, the essential tool used in laboratory to grow, identify and differentiate bacteria. Different bacteria has unique nutrient requirement and respond differentially to their environment. Some need enriched media to grow while others can thrive on basic media. Scientists use selective and differential media to isolate specific organisms and observe their biochemical characteristics. In this video, we explore the most commonly used bacteriological culture, and you will learn what makes each media special and how they work and what to look for while interpreting bacterial growth. By the end of this video, you will have a clear understanding of these essential microbiology tools and how they help identify bacteria in clinical and research laboratories. Let's get started. What is culture media? A culture medium is a nutrient preparation designed to support the growth of microorganisms in laboratory conditions. These media contain essential nutrients such as proteins, carbohydrates, minerals, and growth factors that is required for the microbial growth. Some media also contain pH indicator that change color when bacteria ferment sugars, while selective inhibitors suppress the unwanted organisms. Culture media help microbiologists isolate bacteria, identify species, and differentiate organisms based on biochemical characteristics. First, we will learn about the nutrient agar. Nutrient agar is a basic general-purpose cultural media used in microbiology laboratory. It contains essential nutrients such as peptone, beef extract, and agar to support the bacterial growth. This medium is mainly used for the cultivation of non-fastidious organisms that do not require any type of special nutrients. Because nutrient agar does not contain any selective inhibitors or differential indicators, it allows the growth of a wide range of bacteria. Here in this picture you can see a nutrient agar. The organism is Serratia marcescens. Next is blood agar. Blood agar is an enriched culture media that support the growth of many fastidious bacteria. It contains red blood cells, so it is called blood agar, usually from sheep blood, which allow microbiologists to observe hemolytic activity of the bacteria. Hemolysis refers to the breakdown of the red blood cells by bacterial enzyme. There are different types of hemolysis. Alpha hemolysis produces a greenish discoloration. Beta hemolysis causes complete clearing around the colonies, and gamma hemolysis, which indicate there is no hemolysis. These patterns are important for identifying organisms such as streptococci. Next is MacConkey agar. MacConkey agar is both a selective and differential culture media widely used in microbiology laboratories. It is designed to isolate gram-negative bacteria, particularly members of the Enterobacteriaceae family. The medium contains bile salts which inhibit the gram-positive organisms, and lactose, which is included as a fermentable carbohydrate, and neutral red act as a pH indicator. The lactose fermenting bacteria produce acid, causing the colonies to appear pink in color, whereas the non-lactose fermenters remain colorless. Brilliant green agar is a selective media primarily used for the isolation of the Salmonella species. The medium contains brilliant green dye which inhibit the growth of many gram-positive and gram-negative bacteria. The lactose and sucrose are present as a fermentable sugars, while phenol red acts as a pH indicator. Salmonella typically does not ferment these sugars and therefore produce characteristic colony appearance that help the microbiologist to identify the organisms. XLD agar, xylose lysine deoxycholate agar. It is a selective and differential media commonly used for the isolation of Salmonella and Shigella species from clinical samples. It contains several sugars including xylose, lactose, sucrose along with lysine for decarboxylation testing. The medium also allows detection of hydrogen sulfide production, which appear as black colonies. The phenol red serve as a pH indicator, and bile salts will inhibit the gram-positive organisms. Triple sugar iron agar or TSA agar is a differential medium used to identify enteric bacteria based on the ability to ferment sugars and produce hydrogen sulfide. The medium contains three sugars, that is lactose, sucrose and dextrose. Phenol red is used as a pH indicator to detect the acid production. Some bacteria also produce hydrogen sulfide that forms a black precipitate in the medium. The slant and the butt region in TSA agar provide valuable information for bacterial bacterial identification. Selenite broth is an enrichment medium used in microbiology laboratory for the isolation of Salmonella species from fecal samples. The presence of selenite inhibits the growth of many competing intestinal bacteria while allowing Salmonella organism to multiply. After incubation, samples from the broth are subcultured into any selective agar plate for further identification. EMB agar or eosin methylene blue agar is a selective and differential media used for the identification of lactose fermenting gram-negative bacteria. The dyes eosin and the methylene blue inhibit the gram-positive organism and act as an indicator for lactose fermentation. E. coli typically produce colonies with a characteristic metallic green sheen that you can see in this picture, which helps in its presumptive diagnosis. Edwards medium, it is used for the selective isolation of streptococci. The medium contains esculin which can be hydrolyzed by certain streptococcal species. It also includes red blood cells that allow observation of the hemolysis pattern. Crystal violet and talus sulfate act as selective inhibitors to suppress unwanted bacteria, making it easier to isolate the streptococci. Next is purple base agar supplemented with 1% maltose. It is used for the presumptive identification of Staphylococcus aureus and Staphylococcus pseudo-intermediates. The medium contains bromocresol purple which acts as an pH indicator. When maltose is fermented, acid production causes a color change in the medium which helps differentiate between these bacterial species. Chocolate agar is an enriched culture media prepared by heating the blood agar which lyses the red blood cells and release important growth factors. These include the 10 factor, also known as the hemin, and the 5 factor or NAD. These factors are required by the fastidious organisms such as Haemophilus and Taylorella francisella. Because of its nutrient-rich composition, chocolate agar is widely used in clinical microbiology laboratories.